Hello friends, we are discussing prokaryotic DNA replication. In part 1 of this video, we discuss the first step, that is the initiation of DNA replication. In this video, we are going to discuss about the elongation step of DNA replication. So we know that after initiation, elongation is the next step or the second step in the DNA replication. Now, what is elongation or definition of elongation? It is the synthesis of two new daughter strands complementary to the template strands. Enzyme involved in this step is DNA polymerase 3 enzyme, which is known as the main replicative enzyme. DNA polymerase 1 and DNA polymerase 2 enzymes are also involved. Now, let us see some of the facts of DNA elongation. First of all, direction of elongation. DNA chain elongation proceeds in 5' to 3' direction. Or we can say that DNA polymerase 3 can add DNA nucleotides only in 5' to 3' direction. DNA polymerase 3 cannot initiate the synthesis of new strand, but it requires primer to initiate the strand synthesis. This is because it requires 3' OH group to which it can add nucleotides, and this 3' OH group is provided by the primer, which is synthesized by Prokaryotic enzyme. What are primer? Primers are stretch of DNA or RNA which is 5 to 10 nucleotides long and is complementary to the DNA. In case of the DNA replication, this primer is stretch of RNA. The addition of new nucleotides require energy. This energy is obtained from nucleotides that have 3-phosphate attached to them. Example ATP, adenosine triphosphate. Since elongation or extension takes place in 5' to 3' direction, so there occurs a slight problem at the replication fork. We are aware that DNA double helix is anti-parallel. It means if one strand runs from 5' to 3' in this direction, the other strand runs from 5' to 3' in this direction. New strand, which is complementary to 3' to 5' parental strand, that is this strand, is synthesized towards the replication fork. Hence, this strand is synthesized continuously and is known as leading strand. The other strand, which is complementary to 5' 3' parental strand, grows away from replication fork. It is synthesized in small fragments known as okazaki fragments. It is synthesized discontinuously and it is known as lagging strand. Leading strand is extended by one primer, or for the synthesis of leading strand, only one primer is required. For the synthesis of lagging strand, new primer is required for each okazaki fragments. So this is the parental strand. The two strands are anti-parallel. One has 5' to 3' in this direction and other has 5' to 3' in this direction. This is the replication fork. Helicase enzyme separates the two strands of the DNA. Hence, the replication fork moves in this direction. And the strand which is complementary to 3' 5' parental strand is known as the leading strand. It is synthesized in this direction. 5' to 3' is in this direction and this is synthesized continuously. It is known as leading strand and it requires only one primer. The direction of synthesis is same as the direction of the movement of replication fork. But if we see this strand, this is the parental strand. It is 5' to 3' in this direction. And the new strand or the complementary strand is synthesized in this direction as the nucleotides are always added in 5' to 3' direction. So, the replication fork moves in this direction and this strand is synthesized in this direction, that is opposite to the movement of the replication fork. It is synthesized in the form of the small fragment. And for the synthesis of each Okazaki fragment, one primer is required. So, this strand, which is known as the leading strand, it is extended by one primer, but this strand here, for each of the Okazaki fragment, one new primer is required. So now we know that leading strand is synthesized continuously and lagging strand, it is synthesized discontinuously in the form of the fragments known as Okazaki fragments. We can think or question can come in mind that why lagging strand is synthesized in small fragments or Okazaki fragment. This is because as helicase enzyme separates the two parental strands of the DNA, replication fork moves or grows in this direction. With the separation of the two strands of the DNA, a stretch of template is unveiled or exposed for the synthesis of the lagging strand. Every time only a stretch of template is unveiled, new strand is always synthesized in 5' to 3' direction, or nucleotides are always added in this direction. Hence, a fragment of strand is synthesized because its synthesis is in opposite direction of the movement of replication fork. Then further movement of the DNA B helicase further separates the parental strand. Now again a stretch of the template strand is unveiled and similarly new fragment of discontinuous strand is synthesized. DNA polymerase enzyme detaches from here and attaches here, and then the new fragment is synthesized, and in this way this process is repeated. Since this strand is synthesized opposite to the movement or the growth of the replication fork, and hence it is synthesized in small fragment. So this type of the strand is known as discontinuous strand. But in case of this strand, it is synthesized in the direction of the movement of the replication fork. Hence, it is synthesized continuously. No such problem occurs here. Then again we can think, why is lagging strand so called, or why is lagging strand called the lagging strand and this is known as the leading strand? This is because after a certain stretch of leading strand is synthesized in the direction of the movement of replication fork, around 1000 to 2000 nucleotides in prokaryotes, template of lagging strand is unveiled, and this results in first round of fragment synthesis of the discontinuous strand. Hence, what we see that its synthesis begins after continuous strand synthesis has already started or begun. So, it lags behind this continuous strand. Hence, this strand is known as lagging strand, and the other strand, which is the continuous strand, is known as the leading strand. To continue the addition of the nucleotides, DNA polymerase III is held in place by sliding clamp or known as the beta clamp, which we have already discussed in detail in prokaryotic DNA polymerase video. You can refer it. The link is given in the description box. Also, if any wrong nucleotide base is incorporated by the DNA polymerase III, then this error is removed by the proofreading activity of DNA polymerase III. This proofreading also we have already discussed in general features of DNA polymerase video. You can refer it. The link is given in the description box. DNA polymerase I removes the RNA primer from the newly synthesized strand and replace it with the DNA sequences, because it has both the 5'3' exonuclease activity and 5'3' polymerase activity. So DNA polymerase I enzyme removes this primer and replace it with DNA sequences. Similarly, DNA polymerase 1 enzyme removes the primer of the Okazaki fragment and replace it with the DNA sequences because it has both the 5-3- exonuclease activity and 5-3- polymerase activity. There is nick present between the Okazaki fragment or the different DNA chains. So these are the DNA chains. This is the 5' end and this is the 3' end of this DNA chain. Similarly, 5' end and 3' end of this DNA chain, and so on. This nick is sealed by the DNA ligase enzyme. We will discuss in detail about the DNA ligase enzyme in separate video. This nick is sealed or ligated by forming the phosphodiester bond between the 3' group at the one end of the DNA chain and the 5' group at the end of the other DNA chain, as a result of which Okazaki fragment is joined into complete strand. Now let us see chain elongation reaction that is catalyzed by the DNA polymerase. This chain elongation reaction is nucleophilic attack by 3' group of the primer on the innermost phosphorus atom of the coming nucleotide, deoxynucleotide triphosphate, and this results in the formation of phosphodiester bond or bridge. This is also associated with release of pyrophosphate. So this is the primer, this is the 3' group of the primer. This results in the nucleophilic attack on innermost phosphorus atom of the coming deoxynucleotide triphosphate or nucleotide. This is the deoxynucleotide triphosphate and this is the innermost phosphorus atom. Phosphodiester bridge or bond is formed between the 3'O and the 5' phosphorus atom. This is accompanied by the release of water molecule. And in this way, further nucleotides are added one by one to the growing chain in the 5'3' direction. So let us understand one by one addition of the nucleotides to the growing chain. Suppose there are n number of deoxynucleotide monophosphate, one nucleotide triphosphate comes. So the interaction takes place between them and phosphodiester bond is formed. Now there are n plus 1 number of deoxynucleotide monophosphate in the chain and pyrophosphate is released. In this way, one by one nucleotide comes and attaches to the chain and elongation or growth of the chain takes place. Mg2 plus metal ion is also present in the active site of the polymerase, which play an important role. In the active site of DNA polymerase, Mg2 plus is present. It interacts with this 3'OH and reduces the association between oxygen and hydrogen. This leaves 3'O minus nucleophile, and this 3'O minus nucleophile results in the nucleophilic attack on the innermost phosphorus atom of the coming nucleotide, which then results in the formation of the phosphodiester bond. This hydrogen atom along with OH is released in the form of water molecule, and it also results in the release of pyrophosphate. In this way, one by one nucleotides are added and chain elongation or extension of the chain takes place. So till now, what we have learnt that new strand is always synthesized in the 5' to 3' direction, or in other words, DNA polymerase add nucleotides to the 3' end of a primer or growing DNA strand. So the new strand is always synthesized in this direction, 5' to 3', in this case 5' to 3'. Or we can say that DNA polymerase add nucleotides to the 3' end of the primer or growing DNA strand. We also know that the two strands of the DNA is antiparallel. If one strand has 5' to 3' in this direction, the other has 5' to 3' in this direction. So when these two strands separates and serves as the template for the synthesis of the new strand, then the two complementary strands that are synthesized are also anti-parallel. This newly synthesized strand has 5' to 3' in this direction, and this has 5' to 3' in this direction. Hence what we see that these are synthesized in opposite direction to each other. One of the strand is synthesized in the direction of the movement of replication fork, which is known as the leading strand, and the other is in opposite direction of the movement of replication fork, which is lagging strand. But we have already seen that both the strands are synthesized by the same enzyme, DNA polymerase III enzyme, simultaneously. So how it is possible, as it is not practical to synthesize two strands in opposite direction by the same enzyme simultaneously? The solution to this problem lies in the structure of DNA polymerase III enzyme, in which codomain and beta clamp are present as dimer. And this dimerization is due to the presence of tau, which is the dimerization component. Hence, each codomain and beta clamp is involved in the synthesis of one strand. This codomain and beta clamp is involved in the synthesis of one strand, and this codomain and beta clamp is involved in the synthesis of the other strand. But still there is problem in the direction of the synthesis, because one strand is synthesized in this direction and the other is synthesized in this direction. So at a time, the dimer of the codomain and the beta clamp of DNA polymerase III cannot move in the opposite direction. And this problem is solved by the looping of the template of lagging strand. Since lagging strand is synthesized in the opposite direction of the movement of replication fork. Due to the looping, now what happens? The direction of the synthesis of the Okazaki fragment of lagging strand is same as that of the leading strand. So these two strands can now be synthesized simultaneously in the same direction. And after the synthesis of one fragment of the lagging strand, DNA polymerase III detaches and then attaches at the new point for the synthesis of the next fragment. And in this way, the synthesis of Okazaki fragment continues. This detachment and attachment of DNA polymerase III takes place by beta clamp with the help of the clamp loader, also known as gamma complex. This is all for today's video. In the next video, we will discuss about DNA ligase enzyme in detail. So stay tuned. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.